Hello, everybody. Hope everybody's well. Let's see how we're doing. Starting year 12 tomorrow. Okay, and here because you're bored. Well, this will give you a bit of revision, won't it? Ready for the sixth form. So that would be really useful. Hi, hello. Hi, Lydia. Hi, Zara. Nice to see you. I hope everybody's been having a good summer and probably now back at school, aren't you, most of you? So how's it all going now? Is it weird being back at school? I'm sure the start of term has been interesting, but it must be nice to be back with all your friends. Hi, hello, Ikra. So where is everybody from? Everything's back to normal. I'm so pleased that it's back to normal. It must be a bit of a relief, really. Obviously, we've still got to be careful, but it must be nice. Hi, hello, Magdalena and Maya. Brilliant. So how's everybody doing? Where are you all from? That's the question, because we have people from all over the place. I'm actually not in the Midlands anymore. I'm down in Oxford. Solihull. Yeah, brilliant. Yeah, I was up near Birmingham before, now in Oxfordshire. Right. Brilliant. Poland. Wow. Buckinghamshire, Milton Keynes, you're in the Midlands. There's quite a lot of people relatively close to me. Right, so while we're waiting for people to arrive, what I'll start doing is setting up the sharing of my screen so we can get on with our web class on mitosis and the cell cycle, something that a lot of people struggle a little bit with. And we're hoping that we're going to make it nice and simple so that you fully understand it today. So let's hope that we can iron out any problems that you may have. Please do make sure that you ask questions as we go along, answer my questions if you can. That would be most helpful because obviously the way that I can help you is by answering your questions, isn't it? Okay, Zimbabwe, welcome to Zimbabwe and Jamaica, fantastic. Yeah, we try to cover most exam boards. I'll show you that in the first few slides. We will talk about whereabouts it fits into your syllabus, so don't worry, all right? So yeah, we will talk about where it fits in your particular syllabus so that you can see how it fits in, because hopefully you've got a copy of your syllabus so that you can tick off when you complete things and when you revise things. So you should have a copy of the syllabus from your exam board's website, I hope. So welcome to the GCSE Biology web class. Do you know when we were back? So let's have a look. So we are doing a web class on mitosis and the cell cycle. So welcome to those people who are new. Welcome back to those people who've been to these classes before. Hi, Violet, nice to meet you. So first of all, I'll just introduce who I am. Hopefully you can all see this all right. So I'm the lead GCSE Biology teacher. I've got a BSc in Animal Science, which I did at Leeds University. I also did my PGCE in Biology and Games at Leeds University. I'm a teacher of biology. In fact, I've just finished as head of biology at my last school, but I am carrying on with education writing instead. Hello. So I'm doing content writing now. I hope that you'll find that I have an infectious enthusiasm. I've been an exam marker. I've helped write the syllabus for various different exam boards. And I've written for people like Zigzag Education and CGP, if any of you have come across any of them. So I'm quite experienced on all of that. So please do use my experience to help you understand the topic of mitosis and the cell cycle. So let's have a look at what the objectives are today. Yeah, you can use this for EdXL Biology as well. Yeah, it does. Yeah, it does cover that. So it's very useful for that. All right, so mitosis is covered just about in every syllabus. So what we're going to be talking about today is we're going to talk about the role of the chromosomes in the cell cycle, what happens in each stage of the cell cycle, including cellular division by mitosis. And we're going to explain the importance of mitosis to organisms. What I'm not going to be covering today, because we don't have time, is meiosis, which is the other type of cell division, stem cells and cancer, and culturing microorganisms. We'll cover those in other web classes. All right. So no, not meiosis today. Mitosis today. Meiosis will be covered in another web class. All right. I understand that meiosis is also it's a tricky one, but we've got to get over mitosis first, understand mitosis, and then we'll move on to meiosis. All right. So so that you know the specification points that we're going to be looking at, I'll just briefly mention these, and you can go back and have a look at it after, right? Okay, so mitosis in the cell cycle comes up in AQA, as you can see there. Hopefully you've downloaded the spec from your AQA website. So there's a lot of you, I think, probably doing AQA. Then with OCR, it is describing the process of mitosis and growth, including the cell cycle. And then with EdXL, we're looking at mitosis as part of the cell cycle with the stages of interphase, prophase, metaphase, anaphase, and telophase, PMAT. The importance of mitosis in growth, repair, and asexual reproduction, and how the cell divides. So that's what we're going to be looking at today. So the question is, what do we already know? All right, so what is the process that creates specialized cells? Can anybody remember, you know, when you have your stem cells and then they become specialized cells, such as ciliated epithelial cells or sperm cells or red blood cells? What is that process when they go from that stem cell into the differentiated cell? Can anybody, into the specialized cell? Can anybody tell me what that process is called? Special differentiation, brilliant, to form specialized cells. So it's differentiation. I gave it away then, didn't I? Differentiation gives you our specialized cells from those early stem cells. So how are sex cells? Yeah, so some of you have jumped onto the next bit, which is brilliant. How are sex cells specialized so that they maintain the number of chromosomes in a human body cell after fertilization? So we know that sperm cells and egg cells are specialized, don't we? They've been differentiated, they're specialized, and there's something special about their nucleus. Can anybody remember what's special about their nucleus so that when fertilization occurs, we're not increasing the number of chromosomes? What's special about the nucleus of sex cells? Brilliant. In humans, if we're talking about humans, Kat, yeah, you're right. Body cells contain 46 chromosomes, and our sex cells, the gametes, contain 23 chromosomes. So they, brilliant, Ikra. I hope I'm saying your name right. It's haploid. Yeah, brilliant. So haploid means half the normal number of chromosomes. Well done. All of you have said haploid. So the chromosome number is halved in gametes. So in other words, they are known as haploid, aren't they? Well done for those who got that name. So they have half the normal number of chromosomes. Can anybody tell me what the name is if it's a normal number of chromosomes? It's not haploid, it's something else, which means 2, because they come in pairs. Anybody remember what that's called? So you've got haploid, which is half. Yeah, brilliant. SB. Diploid, yeah, Maya, Cat, Zara, brilliant. Yeah, well done. You're all coming in with your diploids. Fantastic. So haploid and diploid, very important words. They're those key terms that we need to know for our exams, aren't they? All right. They're what get you those marks, those extra marks for knowing your terminology. So what do we already know? Well, I know a lot of you already know this one. So human bodily cells, as we've just said, they are diploid. And sex cells are haploid. So di means 2 because there's 2 of each type of chromosome. All right. And haploid, I always just think haploid half because it helps you, doesn't it? So hello, Mohammed. Good to see you. Somatic cells, excellent. Human body cells have 46 chromosomes. Brilliant. Well done, all of you. You're doing really well. Okay, somatic cells, human body cells. Well done. Right, so human body cells, 46 chromosomes in 23 pairs. So they're in pairs, they're in 2, so it's diploid. 23 chromosomes in the sex cells, in the gametes, which is half the normal number. So that's 23. So that's, in other words, they've got 46 and they have got 23 chromosomes, haven't they? Brilliant. Yeah, and then, yeah, we divide by mitosis. Do gametes also have pairs? No, they don't. The pairs separate, as you will see when we talk about meiosis. So in meiosis, the pairs separate, so only there's 23 chromosomes in each, 1 of each type. Whereas in a normal body cell, you have 2 of each type, 1 from the father, 1 from the mother, and they're numbered 1 to 23. And the 23rd pair are the sex chromosomes, but all the others are the same. So number 1 has the same genes on it as the other number 1. All right. They might not have the same alleles, but they'll have the same genes such as eye color or skin color, etc. All right. Brilliant. Well done. So let's start with part 1, cell division in eukaryotes. So eukaryote means that they've got a true nucleus, doesn't it? Whereas prokaryotes is before a true nucleus, such as bacteria. So we're looking at eukaryotes, which have a nucleus. So we're looking at the cell division in eukaryotes. So first of all, why do cells divide? Why do they need to divide? Has anybody got any idea? The time this session finishes at 3 o'clock. It's a 1-hour session. All right, brilliant. So growth and repair, excellent. So it's growth and development. That's one of them, because the actual definition of growth is a permanent increase in number of cells, isn't it? In size of cells. So growth, yeah, repair, good, growth and repair. Yeah, so replacing old or damaged cells. Replacing, which is repair. Replacing old or damaged cells. Super. All right. So where might cells be actively dividing, do we think? Does anybody know what a lot of dust is, for example? You know, like you have dust in the air. Does anybody got any idea what part of that is, or quite a lot of it is actually? Yeah, hair follicles, brilliant, well done. Yes, there's a lot of division going on in hair follicles. Skin, because we lose skin cells all the time, which is what a lot of the dust is. So skin cells. Yeah, bacteria you do find in the air, yeah, definitely. But a lot of the dust is skin cells, and so we have to replace our skin cells all the time. Human dead skin makes up a lot of dust. You are right, Kat. Yeah. So where might cells be actively dividing? Skin cells, because we've got to replace all those ones that we're losing. Do all kind of cells divide? Pretty much, just at different rates. Some might take years to divide, like some neurons. Some neurons don't divide at all. And then other things like skin cells would divide rapidly. Muscle, because obviously you're growing muscle. So if you are working, you increase your muscle mass, don't you? Any sort of tissues, really. When an egg is being fertilized, it's going to divide because otherwise we're not going to end up with a fetus. So a fertilized egg is also going to be actively dividing. Bone marrow, yes, because you're producing new bone cells all the time, aren't you? So pre-existing cells. So we have stem cells, we have adult stem cells in our body, and they're dividing to replace cells. So we're replacing like liver cells, muscle cells, skin cells, our blood cells. All of those have to be constantly replaced, don't they? And so what we end up with is we have a parent cell like this, and then it divides to form two identical daughter cells. No, muscle cells do divide, have to make new muscle, because otherwise your muscle can't grow. If you think about people who go to the gym and their muscles get bigger, they have to be putting down more muscle cell. All right. And some neurons don't, some neurons do. Adult stem cells are basically undifferentiated cells which divide to produce new cells. So we have adult stem cells in our bone marrow, which will divide to form any type of blood cell, for example. Yeah. So in identical daughter cells, thank you. Yeah, identical daughter cells. Yeah, but you're increasing the number of muscle cells as well. Testosterone is important in muscle mass, yes. All right. So there we go. There's some examples of cell division, and there's so many examples and so many differences between cells as well. And even the types of cells, there's differences between them. So let's have a look at the cell division. All right. So the cell division for growth and repair. What is the name of it? A muscle cell has the contracting protein filaments in it, which brings about the contraction of the muscle. All right. So there's various different muscle cells, and skeletal muscle cell will bring about movement, and you've got heart muscle cell, cardiac muscle cell. Yeah, brilliant. Mitosis. And I always think because obviously meiosis and mitosis are difficult to remember. So I always think growth has a T in it, so mitosis has a T in it as well. Whereas meiosis has an E in it, and it's for sexual reproduction, and sex has an E in it. Okay, brilliant. So the parent cell, do you remember we were talking about haploid and diploid? What is the name of the number of chromosomes in a body cell? Is it haploid or diploid? So we're looking at the parent cell here. This is the normal body cell. Is it haploid or diploid? Remembering about how I taught you to remember the differences between, yeah, brilliant. Well done. Yeah, you're all coming up with it. So the parent cell is a diploid cell, and it wants to produce identical body cells. All right, so it wants to make identical cells. So do you think these daughter cells are going to be haploid or diploid? It's a slightly different process. Binary fission refers to bacteria which don't have nuclei. Yeah. Oh, yeah. The daughter cells are going to be identical to the parent cell. So if they're identical, they have to have the same number of chromosomes. Yeah. So they're still diploid, because we want to end up with identical cells, because they're just replacing the parental cell. So that's why they need to be diploid. So it's haploid if we're making gametes for sexual reproduction, but mitosis is all about growth and repair. So we want to make identical cells, don't we? So they have to have the same number of chromosomes. So their diploid parental cell has to produce diploid daughter cells. all right? Haploid is involved in producing gametes for sexual reproduction, and that's a different cell division. And this is why people get confused, all right? So haploid daughter cells come out of meiosis, which is the production of gametes for sexual reproduction. We're talking about mitosis, which is for growth and repair. And so therefore we're producing identical cells to the parental cell. Hopefully that makes sense for those people who got a little bit confused there. All right, so this is all about the production of body cells, not gametes, okay? So adult cells, right, the adult stem cells are undifferentiated cells which can divide into a limited range of new cells, so, and differentiate into a limited number of new cells. So like in bone marrow, there are stem cells which will divide to form any of your blood cells, all right? So adult stem cells can divide to form a limited number of different types of cells. So a lot of our new cells come from adult stem cells. All right, so how are we feeling? How confident are you feeling? Straightforward? All right, super confused? What do we reckon? 1, 2, or 3? Let's see how you're doing. How are we feeling? 1, 2, or 3? 1, 1, 1, good. Oh, super duper. Yeah, excellent. Right, so let's have a look at a couple of exam questions, shall we? Brilliant, I love the happy face. Brilliant, so explain why DNA replicates before cells divide. Remember what I was saying about the parental cell wanting to produce identical daughter cells, all right? And the fact that we're going from diploid to diploid. So why do we think that DNA has to replicate before a cell divides? So this is actually an OCR higher paper. Yeah, it needs DNA for both cells, fantastic. So we need the same amount and the same DNA for each cell as the parental cell, don't we? So the same, so there is DNA or genetic material, it doesn't matter, in each new, ooh, that didn't quite come out as new, did it? A new cell after division, because we want identical cells to that original parental cell, don't we? All right. Yeah, so we've got to double that DNA before we split it into 2, because we're going to have to halve it again, aren't we, to make 2 new cells. So we've got to double that DNA at the beginning so that we can then halve it and we still end up with the same number of chromosomes at the end as we started with. Super. That's a really good explanation, some of you. Really good. Well done. Right, so let's move on then to, well, should we move on to the cell cycle and mitosis then? Let's look at a little bit more detail about what actually happens in the cell cycle in mitosis. To start off with, what is a cell cycle? So we have to start with the parental cell and we have to end up with 2 identical daughter cells, don't we? So what we have to do is we're going to split it down into 3 stages, basically. All right, so we've got stage 1, stage 2, and stage 3. So what can you see as the difference, all right, between this cell here and this cell here? What's the obvious difference that's happening in stage 1? Can anybody tell me? Yeah, brilliant. The cell cycle is a series of events as it grows and divides. That's perfect. And you've mentioned it already right at the beginning. It grows and divides. Yes. So this part is called interphase because it's actually between the splitting of the cell. So it's the phase between the actual splitting. So this is known as interphase, and the cell grows. Brilliant. Everything is replicated. Good. So the organelles have replicated, haven't they? The chromosomes have doubled. Well done. So chromosomes double in number. And we increase the number of organelles. Because each cell needs their own organelles. They're not going to be able to survive if they haven't got enough mitochondria, for example, to release energy. So everything is replicated. The chromosomes have multiplied, and yep, G1 cap. So everything has multiplied. The cell has grown because it's got to get bigger because it's going to be split into two. We've got to have those mitochondria. We've got to have those ribosomes for each cell, because otherwise they're not going to be able to carry out all of the things that they need to live. So interphase is all about that growth and replicating, ready for that division. All right, so this is stage 1, interphase. All right, so with the replication of the DNA, obviously the DNA, as we know, is a double helix, and it zips apart, and then new strands are formed to make this new DNA. All right, so they use it as copying. We'll go into that in more detail in another lesson. So what is the cell cycle? Let's look at stage 2. What's actually happening in stage 2? What's happening to the chromosomes? What has happened to the chromosomes? Instead of being here in the nucleus, what's happened to them? Yeah, so mitosis, excellent. So what's happened is those doubled up chromosomes have divided, haven't they? They've gone to opposite ends of the cell. Yeah, so we go through this, mitosis goes through prophase, metaphase, anaphase, telophase, doesn't it? So we'll look at that. So mitosis involves prophase, metaphase, when they line up in the middle of the cell, anaphase, where the chromosomes move away from each other, and telophase, when the new nuclei form, and then we go into cytokinesis. So mitosis, all right. And stage 3, does anybody know what it means, what the word is for saying that the cell has split into 2? So mitosis is PMAT, yeah. Brilliant. So the splitting, stage 3, when the cell actually divides into 2, is known as cytokinesis. Cyto means cell. Kinesis means to split. All right, so cyto, cell, splitting. So stage 3 is when they actually divide into 2. So that's cytokinesis. Excellent. So now let's have a look at that in the mitosis part in a little bit more detail. So we've got the idea of the cell cycle going through the growth part, the replication, the dividing of the chromosomes, and then the splitting of the cell. So that's the whole cell cycle. But obviously we've now got to look at mitosis itself, haven't we? All right. So let's have a look. Okay, so this bit is going to be a little bit, so we're going to spend a little bit of time discussing this, this slide. We'll stay on here for a little while just to discuss it in a little bit more detail. So this is mitosis, all right. So what we've got to start off with, all right, this is the nuclear membrane. In other words, it's the membrane which goes around the nucleus, and inside you can see that there's 4 chromosomes, can't you? All right, so in this first stage we're going from here. Let's look at what's happened from this first one to here. What's happened in this arrow A? What's happened to A, the nuclear membrane, and what's happened to the chromosomes? I need both of that. Looking at that diagram, what has happened to both the nuclear membrane and the number of chromosomes. Yeah, so the nuclear membrane has disappeared. The chromosomes, which are now doubled in number, also become more visible, all right? So the doubled number of chromosomes from interphase condense, so we can actually see them properly. And so this one is a copy of this one. All right, so what we're doing is we're making identical copies, but holding them together so they don't get all jumbled up. All right, so the doubled number of chromosomes then condense. G0, yeah, so that's in between growth when there's no growth, I believe. Is this for prophase? Yes, this is prophase. All right, this stage that we're looking at the moment is prophase. All right, good. So this links into prophase, because some of you need to know about prophase and some of you don't. All right, so that first stage, the first phase, prophase, the beginning phase, that's what pro means. So prophase means the beginning phase, and what's happened is those chromosomes which have doubled up condense. What they do is they coil really, really tightly. And why they coil really, really tightly, that's what they condense means. They're coiling really, really tightly around these little proteins down the middle called histones. What they do is, because they coil tightly, it means that they don't get tangled up. Because if you think about, if you have string, you're not just going to dump it in your drawer. You're going to roll it into a ball so that it doesn't get tangled up. That's exactly what those chromosomes are doing. Instead of just dumping the string in the drawer, all right, what they do is they tightly coil them around these histones around the middle, like we would coil string around a cardboard tube to stop it all getting tangled up. The chromosomes condense, they wind tightly around these histones so that they don't get tangled up. And that means that they're going to be able to divide easily without too much tangling up. Yeah, so DNA is copied in interphase. What we're showing here is that you can see now that there's two distinct chromosomes because they've coiled up. All right, so the doubled number of chromosomes condensed. Does that make sense? So now we can see them as two distinct chromosomes. They're just held together at the middle cap. Is that all right? Yeah, the resting phase when there's no growth. Exactly. Okay. So what comes after prophase? Those people who are doing prophase, metaphase, anaphase, and telophase. Yeah, that's right, Kat. Okay. So they condense around the histones in prophase. So now we're going on to metaphase. Only a few of you need to know about this. I think it's just Edexcel need to know about this, but you may have been taught it anyway, because a lot of teachers teach it in these stages because it's easier to explain it in the stages. So this is metaphase. Now I always remember M for middle. Metaphase, M for middle, because what's happening are those pairs of chromosomes are lining up down the middle of the cell. Yeah, I'm pretty sure it is at AQA. It's just that some of you have learned it, and for the Edexcel people, you need to know it. If you don't need to know it, it doesn't matter. All you need to know is what's happening. All right. Yeah. Right, in metaphase, the two centrosomes are not actually moving to opposite ends yet. That comes in anaphase. All they're doing is they're lining up down the middle. All right, so metaphase, M for middle. So the what they're doing, as you can see here, is that they're lining up what we call around the equator of the cell. So we've got this thing called a spindle, which some of you will have heard about. Basically, it's these fine fibers which are laid down between the poles of the cell, the opposite ends of the cell. And those chromosomes go and basically stick to those fibers. And they're in their pairs and they're lined up along the center of the cell. And what's going to happen is one's going to go one way and one's going to go the other way, because they're going to be pulled apart by those fibers. So those fibers are quite important that make up the spindle. So what they're doing, yes, they do. They are between the spindle. Well, there's the spindles in some cells, not in others, but it's not always between the centrioles, but the spindle fibers are laid down between the poles from top to bottom. Yeah, don't worry too much. If you don't need to know about centrosome, centriole, and centromere, don't worry about it. Just remember the pairs of chromosomes line up across the middle of the cell. All right, that's all you need to know to get through your GCSEs pretty much, okay, unless you're with Edexcel, and then you need to know prophase, metaphase, anaphase, telophase. So all you need to know are those pairs of chromosomes line up across the middle of the cell. So don't worry about all those different words. A level, if you're going to do A level, you will need to know about it, all right, but if you're not going to do A level, you don't need to know about it. So the pairs of chromosomes line up across the middle of the cell. That's all you need to know for most of you. The pairs of chromosomes line up across the middle of the cell. Okay? Were you all happy with that? Are any questions on that part? Right, so stage 3 then. They are moving away from each other. So those people who are doing Edexcel or who've learnt the stages, what stage is this? Yeah, brilliant, anaphase. And again, A for away, because what's happening is those pairs, the fibers are pulling them in opposite directions. So they basically, the fibers split and pull from the poles of the cells, and they pull these pairs of chromosomes apart. All right, does that make sense? So you can actually see this one's going this way, this one's going this way, this one's going that way, that one's going that way. So they're just getting pulled apart. So they separate where they were glued together in the middle. All right, they separate, and this means that we get the exact copies of chromosomes going to each side, because remember, these are exact copies of each other, aren't they? So we're going to end up with exact copies at each end of the cell. Yeah. It's the sister chromatids which make up that pair. So you've had this replicated pair. You had the original chromosome, which made an identical copy of it. The identical copy gets pulled one way and the chromosome gets pulled the other way. Does that make sense, Kat? The centromeres separate, the spindle fibers shorten, and chromatids are pulled by their centromeres to opposite poles. Absolutely. Yeah, that is completely correct. All right. So basically what's happening, we've got those identical pairs. One gets pulled one way, one gets pulled the other way. That's all that matters. All right. So the chromosomes, the identical chromosomes, are pulled to each end of the cell. So now what we've got, those are identical to those, which is brilliant. That makes life easy, because now we are going to end up with identical cells. All right. So now what we've got is we've got these identical, those are identical to these. Okay. So we go into our last part of mitosis, all right, where we are in telophase. And what we've got is the cytoplasm and membranes divide. And then we've got new nuclei formed, don't we, in this last one? Because now what we've got, can you see here, we've got the nuclear envelope. And this happens at the same stage as the actual cells dividing into two, so which is cytokinesis, of course. And what happens in animal cells, it's like pinching blue tack into two, when they get pinched into two and they get pulled apart. In plant cells, what happens is a new cell wall has to be formed and then they split apart. All right. So there is a difference with cytokinesis between animal cells and plant cells. So with animal cells, they pinch into two, plant cells, a new cell wall is laid down. Yeah, in AQA GCSE, you are allowed to write about prophase, metaphase, anaphase, and telophase. They're not going to penalize you for having additional knowledge. So don't worry about that. If it makes sense to you, because I know a lot of my students prefer it when they go through PMAT, it makes more sense to them to go through that. So that's absolutely fine to talk about those different stages, even if it's not on your syllabus, because they can't penalize you for knowing more than the syllabus asks you to. Yeah, that's right. Kat, in the animal cells, there's a contractile. It's like the fibers of the spindle. They're microtubules which go around the center of the cell, and they tighten and basically pinch the cell into two. All right, so that's really good. Well done. All right. No, it doesn't. No, every exam board just has different extras that they put in, Maya. So EdXL will be harder for some things and they won't need to know as much as AQA on something else. And it's just how they've chosen to write the syllabus. They've decided they want to put PMAT in ready for A-level, possibly. It just depends, but they all equal out pretty much the same. It's just each different board choose different detail. All right, so hopefully that makes sense to you. So let's have a look. How confident are you feeling on this part? It's a little bit trickier, isn't it? But hopefully it makes sense to you. Yeah, 1, brilliant. This is good, it's looking good. EdXL and A-level are the same? No, it's just that EdXL have chosen to do prophase, metaphase, anaphase, and telophase, and other exam boards haven't. But other exam boards will go into a little bit more detail on something else. So it's just one of those things, okay? How hard is A-level biology? Well, that's a good question. It goes into a lot more detail than GCSE. So if you find GCSE okay and you understand it, you should be all right with A-level biology. But yeah, it's a lot harder. A lot of people think that A-level biology is an easy science, and it isn't. You're doing it, are you? You've just started. Yeah. You'll know in the first few weeks how you're feeling about it. You got an 8, you should be fine. All right, you should be fine with it. So it's just, you just got to keep on top of the volume of work, because A-level has got a huge volume of work. All right, because as you know, biology has a lot of content in it. There's a lot of written work, isn't there? Okay. Right, let's have a look at an exam question then. This is a 3-marker. So what we're looking at here is mitosis occurs in plant cells during growth. Yeah, definitely a lot to learn in A-levels. Yeah, unfortunately, you just got to go in with that mind that I've got to keep my head down and don't get behind with your work. If you get behind, there's no catching up. Basically, it's very hard to catch up anyway. Just keep on top of it and you'll be all right. Okay, you've just got to keep working. It's a big step up from GCSE, no doubt about it. But you can do it. If you're determined, you can do it. All right, it's like going on to university after A-level. These are all big steps. So let's have a look at this then. Mitosis occurs in plant cells during growth. So we know we're looking at mitosis, all right, where we're producing identical daughter cells from the original parental cell. It wants you to describe the division of a cell by mitosis. All right. So we are basically looking at the mitosis part, aren't we? So we're looking, for those people who like the prophase, metaphase, anaphase, telophase, we're looking at that. Or we're just thinking back to those diagrams we've just done when we went through each of those stages. All right. So we've got to basically put those pictures into your head, think about them and think how we're going to explain it. So what happened in that first picture? Can anybody remember what happened in that first picture? What did we talk about for prophase, or that first picture? What happened to the nuclear membrane, for example? Can anybody tell me? Yeah, so in prophase, what happened? What happened to the nuclear membrane? Let's start with that. Easy start. And the chromosomes doubled. The chromosomes double in interphase, don't they? What happens to those doubled chromosomes? Good. So the nuclear membrane disappears. We're going to write down more than 3 points, by the way, but any 3 of these are going to be fine. So nuclear membrane disappears. What happens to those doubled chromosomes? Brilliant. Breaks down. What happens to those doubled chromosomes? Can anybody remember what I talked about with string? Yeah, the chromosomes. So the doubled chromosomes condense, don't they? So we can now see them as pairs, identical pairs, sister chromatids, but the identical pairs. Brilliant. So that's the first stage. Now, can you remember what happened at the next stage? Those people who remember it as metaphase, but the M stood for something, even if you don't want to remember metaphase. What did the M stand for? What happened to those pairs of chromosomes in the next stage? The M meant something. Can anybody remember what happened at that next stage? Yeah, brilliant. The pairs of chromosomes line across. Brilliant. Super. Brilliant. So now we're going to go into the anaphase bit. And if you're not doing anaphase, it doesn't matter. What does that A stand for? Can anybody remember what A stood for the next bit? So we prepared the chromosomes in prophase. They're down the middle in metaphase. What happens with the A? Chromosomes separate. Brilliant. So the chromosomes are pulled apart. Okay, so they've been pulled to the opposite side of the cell. Now what happens, because we've obviously got identical chromosomes at each end. So what's going to happen now? What forms around those chromosomes? Now, Hamid, if you go to AQA website, you should be able to download the spec there. And that will be really, it's so useful to have it in your folder. Yeah, nuclear envelope reforms, nuclear envelope or nuclear membrane, whichever you want to call it. So you can also call it envelope because it's a double membrane. Yeah, and they decondense. I like that one. They unravel, decondense. Yeah, so we've got the formation of the nuclei So we've ended up, and then obviously it goes into cytokinesis. So what do we end up with at the end? What is the final product of mitosis? I know it's hard. That's why we're trying to dedicate a whole hour to mitosis. Anything you want to ask at the end, we've got enough time. We've got 10 minutes left of the web class. So anything you want to ask, just ask and I will explain it. All right. So cytokine separates and we end up with two genetically. That's a really important thing to include in your answer. They're two genetically identical diploid daughter cells. Now we've got lots of good words in there, haven't we? Because remember, you've got to use those keywords to get those marks, haven't you? Yeah, the cleavage furrow. Yeah, so the cleavage furrow forms on animal cells. We get that pinching into two, don't we? And in plant cells, but we're talking about plant cells. So in plant cells, what happens? Okay, here's your cell wall around the original cell. And now we've got two new nuclei, haven't we? What happens is a cell plate forms like that across the cell, and then a new cell wall forms on that side, and a new cell wall forms on that side, and then they can split into two. All right. So the cleavage furrow forms in animal cells, because what happens with animal cells, it goes like that. All right. And so it pinches into two, a bit like you pinching plasticine or blue tack into two. But with plant cells, we have to lay down what we call a cell plate, which then you form your cell wall onto. Yeah, it is the cell plate formation, and then the cell wall builds on that. Brilliant. Well done, Kat. That's a lot of detail that you're going into there, Kat. That's really good. How do you remember what happens at each stage? Well, I do like using PMAT, whether you use prophase, metaphase, anaphase, or telophase or not, it doesn't really matter, because this is preparation. So that's preparation. So you're trying to make sure that those chromosomes don't get tangled up, and that's why they condense. That's why they, and if you can't remember condense, you just say they coil up really tightly, and that's so that they don't get tangled up. All right. M, so that's preparation. M for middle, A for away, so the chromosomes move away from each other, okay, and this T for two, because we're now forming two new nuclei, two new cells. Yeah, okay, so that's how a lot of people remember it is PMAT. I hope that helps a little bit. So you've got preparation, where you're stopping them from tangling by wrapping them up around the histones, coiling them up. M for middle, A for away, T for two. All right. And if you can try to remember that, you won't be far off. All right. Because then you've got the most important points for each one. All right. So there we go. So are there any more questions? Because we've now come to the end of there. So what we should now know is the role of chromosomes. So chromosomes in the cell cycle are getting doubled so that they can be passed on to the new cell, aren't they? Because we need to have the exactly same genes going to all of the cells so they can all carry out their functions that they should do. Describe what happens in each stage of the cell cycle. So we had interphase, then we had mitosis, then we had cytokinesis, didn't we? All right. So interphase is between the splitting. Okay. Then we've got the mitosis, which is your PMAT. And then you've got your cytokinesis, cytocell kinesis splitting. All right. So we've looked at the cell cycle. Then we looked at mitosis in more detail, the different stages, anaphase and telophase, photo of a cell and ask you to identify it. Right, anaphase is literally when you are seeing them going like that away, all right? So when they're moving away like that, that is anaphase, and it could be anywhere from here to up there. Then in telophase, what happens is that you'd have them all round like this, because what they're going to do now is they're forming the new nuclei. Does that make sense? So if you're looking at a picture, it's all about, are they physically moving away from each other? That would be anaphase. If they're in a bundle at each end, then we've got telophase. Hopefully that makes sense, Kat. All right. Any other questions on that? Any more questions for me? I know it's a complicated topic, but hopefully it's helped you a little bit. But we've still got a few minutes, so if you've got any questions for me. Just to remind you, we still have some more web classes coming up. So if you are doing all of the web classes, and I hope you are going and trying them all out, there's the physics web class is coming up on the 19th. We've got, obviously that's slightly different because we've done a little bit of a rejig. And then there's maths, there's chemistry, and I'm back on biology with digestion, enzymes and metabolism on the 17th of October. So I hope I'm going to see some of you. The nucleus and nucleolus. The nucleus is surrounded by the nuclear membrane. So that's all of the different components and all the DNA. And the nucleolus is where the DNA is concentrated. Interphase, what's happening in interphase is you've got growth of the cell and you've got replication of the organelles, and replication of the chromosomes is happening at that stage as well. It's basically preparing, it's making sure that you've got all the components ready for cell division to occur. All right. Because it can't divide if it hasn't got double of everything, basically, because otherwise those cells, those daughter cells, are not going to be able to carry out their function. So there we go. All right. It's where RNA is also produced, not ribosomes as such, RNA. Okay. So GCSE maths on the 26th of September, everybody, at 3 p.m. Okay. Right, well, it was lovely to see you all, and I hope to see you all again soon. Have a good week at school, and I'll see you in October, ready for enzymes. Okay, take care, everybody. Have a good week. I'm glad I've been helpful. Thank you. Excellent. Okay, take care.